My name is Andre Martel from the Canadian Museum of Nature. This talk on the discovery of the Eastern Pearlshell mussel Margaritifera and the fish community in two tributaries of the Ottawa River is co-authored by Jessica Reed, Noel Alfonso, Annie Paquet, Sophie Imprich, Jacqueline Medill, Katrina Ilves, Leanne Noel, Jürgen Geist, and Stephen Cook. There are species of freshwater mussels that, in order to thrive and be abundant, are known to require very specific habitats, a very special type of stream or river, and on top of that, they require a very specific host fish. The Eastern Pearlshell, also called freshwater pearl mussel in Europe, Margaritifera, is one of those species. It thrives in healthy streams and rivers with crystal clear water, low silt, oxygenated cool water, high quality substrate, presence of salmonids, and the species has often been labeled, and with good reasons, an ecological indicator or a flagship species, and its conservation biology and ecology has been extensively reviewed in previous studies. The general distribution of the Eastern Pearlshell mussel in northeastern North America is seen here in the shaded blue oval. Basically, it's a vast area that includes rivers and streams with populations of Atlantic salmon or brook trout, or both, salmonid fishes being the host for this mussel. This talk is about the discovery of a population of Eastern pearlshells in the Kenauk River, shown here at the end of the pointed line, in the Ottawa River watershed, an area that is much further inland, much further west than the shaded blue oval distribution area shown on the previous slide. This is so far Canada's westernmost population of this species. The Kenauk River is located in a pristine wilderness area of 263 square kilometers, owned by the Kenauk Institute and in part by the Nature Conservancy of Canada. This wilderness area, unique in Canada, has been privately owned since 1674. Most of the watershed is within Kenauk, so privately owned. During this talk, I will be talking about the Kenauk River and the West Kenauk River that you see here to the left of the slide with arrows pointing. The discovery of a population of the Eastern Pearlshell mussel in the Ottawa River watershed at Kenauk led to the conducting of a multidisciplinary study. And for this talk, I will go over very briefly through some of the key results our team has obtained. This is the plan of the presentation, and I want to mention at this point that for items 3 and 4, these are parts of the thesis of Sophie Imprich in Jürgen's lab on substrate quality and recruitment, and of Jessica Reed in Steve Cook's lab on fish surveys. First, let's talk about the extraordinary untouched riparian forests surrounding the Kenauk and West Kenauk rivers. As you can appreciate on this picture, the riparian forest is made of old-growth trees, including century-old white cedars. This picture was taken at the site of the discovery of the species in September 2018. According to locals and historical archives, the riparian forests bordering the valleys of the Kenauk River and West Kenauk River have never been logged. The Kenauk River is a mid-size river with low to medium gradient and water flow. Very healthy riparian zone, never been cut, as we said before, and warm water in the summer, 24 to 26 centigrade in July, August. Number 2, the distribution of pearlshell mussels. A quadrat of 1/4 meter square was used, as well as the method of aquascope and snorkeling. The red dots on this map show you where live Eastern Pearlshell mussels were found. In the West Kenauk, the density of population between 5 and 150 individuals per square meter, very high density of Eastern Pearlshell in the West Kenauk, and it was the only freshwater mussel species found. In the Kenauk River proper, more to the right, very low density, usually less than 1 individual per square meter, and the Eastern Pearlshell was also found along with other species of freshwater mussels, the Eastern Elliptio, the plain pocketbook, and the triangle floater. It is now recognized that the quality of the substrate is crucial for the survival of early juveniles of pearl-shell mussels. And papers by Jürgen and his colleagues in Europe have clearly shown the importance of examining parameters such as turbidity, oxygen, and interstitial water flow, as well as the redox value, to evaluate within the first 5 centimeters of substrate and measure the quality of the substrate for pearl shells. And this is what Sophie did at Kenauk for part of her thesis. This table summarizes some of the physical chemistry 5 centimeters within the substrate in the interstitial space for the Kenauk River, the red values, and the West Kenauk River, the blue values. If we just examine the mean for dissolved oxygen and water temperature, the values you see in the blue oval. The West Kenauk, the blue values, has on average more dissolved oxygen and lower water temperature within the substrate compared to the Kenauk River, the red values. The key message from this table is a higher quality of habitat for juvenile pearl shells in the West Kenauk. So within the quadrats that we were examining and during Sophie's thesis, finding juveniles within the substrate buried in the sediment was a clear confirmation, especially when you find small ones like this, like the ones in the center and towards the right and in the hands here in Sophie's hand. Clear confirmation that the population is recruiting, that there is juvenile recruitment. So that's a functional population, and this is what we saw, should I say, in the West Kenauk and not in the Kenauk. Number 4, fish surveys, including the brook trout. The method that was used was electrofishing, and that was part again of Jessica Reed's thesis, working with Steve Cook at Carleton University, and Jessica, assisted by Noel Alfonso, Katrina, via Noel. The work was done in the Kenauk and West Kenauk, and again, it was a catch and release approach using electrofishing. If we look at the fish diversity for both the West Kenauk and main Kenauk, we have 21 species in total, and the West Kenauk had 9 species. If you look at the bottom, you can see a total of 9 species for the West Kenauk, and for the main Kenauk, 16 species. A key point to make here is that the brook trout, the presumed host fish of the pearl shell, was present when Jessica was electrofishing in the West Kenauk, was present in the West Kenauk, but was absent in the main Kenauk. Also note the smallmouth bass, also present in the main Kenauk, which is a larger river, warmer waters, and the bass is present, but there's no smallmouth bass in the West Kenauk. One thing important to mention, no Atlantic salmon has ever been reported at Kenauk. So using the fish data and knowledge on the fish diversity in the system at Kenauk, with no Atlantic salmon, so crossing Atlantic salmon here on top, which is such a common host fish for pearl shell in Atlantic Canada and in New England. So we're left with the brook trout as the presumed host at Kenauk. And this is part of our future research to confirm the presence of Glaucidia on the gills of this fish. So comparing the two rivers, our study shows that the West Kenauk is an ideal place for both the Eastern Pearlshell mussel and the brook trout. The difference between the West Kenauk functional population and the Kenauk River non-functional population of pearl shells led us to the following question. Could the West Kenauk River occasionally supply the main Kenauk River of juvenile pearl shells via brook trout with Glochidia on their gills, descending the West Kenauk and spreading upstream and downstream in the main Kenauk River? We don't have an answer to that question. Number 5, the genetics of pearl shells from Kenauk. This neighbor joining phenogram based on the genetic distance of 26 Margaritifera populations from across North America, with 2 outgroups from Germany at the bottom left, DZ1 and DWB. So it shows, based on 9 microsatellite loci, it shows how different, the red circle here shows the Kenauk pearl shells, how different they are from the rest of the North American populations. Much of the data on this phenogram on the upper left portion of it is from David Zanata's paper, who worked with Jürgen and other collaborators to examine the genome of pearl shell populations across North America. So the key point to remember is how the Kenauk population is different from the rest of the North American pearl shell populations. Here is another way to look at the difference in terms of the genetics of the Kenauk pearl shell mussels. In red, to the right of the diagram, you can see in the oval there how different each dot, each individual mussel examined is from the rest of the North American populations in the middle of the diagram there. So each dot, its color indicates how similar or how different an individual is in terms of its genome based on the 9 microsatellite loci. The 2 leftmost groups are from Germany as odd groups. Look how different they are. The key point being for this figure how unique the Kenauk pearl shell mussels genetics is in terms of microsatellite loci examination compared to the rest of the North American populations. So number 6, conclusions to wrap up. Pearl shells at Kenauk, westernmost population in Canada. The West Kenauk, an ideal river for pearl shells, high density of population. Juveniles are into the sediment. Functional population, evidence of recruitment. Excellent substrate quality. Cooler summer water temperature and wild brook trout present. The Kenauk, the main river, low density of population only. No small juveniles detected in the sediment. Therefore, non-functional population. Substrate quality is good, but warm water in the summer. Brook trout is absent, and the smallmouth bass is present. Genetics. The Kenauk pearl shells form a distinct group from all other North American populations. Conservation of pearl shell population at Kenauk is linked to the conservation of wild stocks of brook trout and the conservation of the vast untouched riparian forests. This study is also a confirmation that the Eastern Pearlshell is a good ecological indicator. Functional populations of this mussel are associated with pristine streams or rivers, with low silt, cool water, high quality substrate, and presence of salmonids. This study is also in strong support for using Eastern Pearlshell mussels as target species in conservation effort. So this is the last slide for this presentation. I thank you for listening.